Dear students, today we will discuss about food fermentations part 4, fermented vegetables. Vegetables are preserved by fermentation from time immemorial. A wide range of vegetables and fruits including cabbages, olives, cucumber, onion, pepper, tomato, carrot, celery, cauliflower, etc. are preserved by fermentation. The fermentation for vegetables results in the formation of nutritious foods and they will have an extended period of storage time. Before fermentation, the fruits and vegetables may be containing a variety of microorganisms including spoilage bacteria, fungi, etc. and the growth of these microorganisms deteriorates the vegetable material due to the action of degradative enzymes like protease, lipase, amylase, etc., which are released from these microorganisms. The fermentation of vegetables results in the production of organic acid, carbon dioxide, and a variety of antimicrobial compounds by lactic acid bacteria. As a result, due to the production of acid, it lowers the pH, which inhibits the development of the spoilage microorganisms and their enzymatic activity. Also, the carbon dioxide produced during the fermentation will provide anaerobic condition which favors the stabilization of vitamins as well as it will stabilize the natural color of vegetables. The vegetables are fermented through either of the following methods: dry salted, brine salted or non-salted. In dry salting method, the vegetable is treated with dry salt and due to osmosis the juice from the vegetable comes out and it will form the brine. In brine salted, the vegetables are kept in a brine solution containing 15 to 20% salt. In the non-salted method, the vegetables are fermented by lactic acid bacteria without the addition of salt. What happens during fermentation? In the fermentation of vegetables. During the fermentation of vegetables, the glucose and fructose present in the vegetable will be converted to lactic acid, acetic acid, ethanol, carbon dioxide, etc. by the action of lactic acid bacteria and yeast. There will be homofermentative lactic acid bacteria which results in the formation of lactic acid, and the growth of heterofermentative lactic acid bacteria will result in the formation of carbon dioxide, lactic acid, ethanol, acetic acid, etc. Now we will discuss about different vegetable fermentations. Coming to cabbage fermentations, sauerkraut. This is an example for the commercial production of fermented cabbage, mainly in the United States and Europe. To prepare sauerkraut, cabbage is sliced into thin pieces which are known as sloe, and this sloe will be immersed in 2.5% salt at a temperature of about 15 degrees Celsius. During this period, the sloe must be completely immersed in the brine to prevent it from darkening. The initial fermentation is carried out by leuconostoc mesenteroids, which is a heterofermentative lactic acid bacteria. There will be formation of acid and carbon dioxide, which will create an acidic anaerobic condition which will eliminate the growth of other microorganisms. As a result, the growth of other lactic acid bacteria will be favored, and there will be the growth of Lactobacillus brevis, Lactobacillus plantarum, Pediococcus cerevisiae, etc. Other bacteria such as coliforms, pseudomonas, etc. will disappear. The growth of the lactic acid bacteria will produce several compounds such as lactic acid, acetic acid, ethanol, volatile compounds such as diacetyl, acetaldehyde, acetal, isoamyl alcohol, hexanol, ethyl acetate, ethyl butyrate, isoamyl acetate, etc. And these molecules will contribute to the flavor of sauerkraut. Generally, sauerkraut fermentation are done in large fermentation tanks in which the shredded or chopped cabbage known as sloe is kept in brine. The salt during the fermentation of cabbage serves several purposes. It extracts moisture from the cabbage by osmosis to form the brine in which the fermentation will take place. Salt inhibits some of the natural spoilage microflora of the cabbage and helps the selective growth of lactic acid bacteria. It also helps to maintain the crisp texture of the cabbage by withdrawing water and also by inhibiting endogenous pectinolytic enzymes. Salt also contributes to the flavor of the product. The next fermented vegetable is kimchi. This is also a cabbage fermentation usually used in Korea, made from Chinese cabbage. The kimchi fermentation is microbiologically similar to sauerkraut fermentation, except the ingredients, flavor, and the preparation methods. For kimchi fermentation, the cabbage is cut and soaked in a brine of 5 to 10% NaCl. Then the cabbage is washed and drained, and an aqueous paste of ground red pepper is mixed with the cabbage. Small amounts of additional ingredients such as garlic, ginger, and geotkel and green onion are also usually added. Geotkel is a high salted anchovy fish. The kimchi fermentation also have a biphasic heterofermentative and then a homofermentative pattern of lactic acid bacterial growth. The prominent species of microorganisms involved are Leuconostoc misandroids, Pediococcus cerevisiae, Lactobacillus brevis, and Lactobacillus plantarum. After their growth, the final salt concentration will be between 3 to 6%. In rural areas of Korea, kimchi is traditionally fermented in earthen jars. And in urban Korea, kimchi is prepared by using household kimchi refrigerators. More than 65 different types of kimchi are identified based on their differences in raw materials and processing. During cabbage fermentation, both in kimchi as well as in sauerkraut, vitamin B content will increase and the vitamin content of vitamin C and A are preserved. Next, fermented vegetable is olive fermentations. There are several methods for fermenting olives. They are green table olives, natural black olives, and ripe black olives. First, we will discuss about green table olive. So here, the green olives are treated with lye, which is 1 to 3% sodium hydroxide. The sodium hydroxide treatment will reduce the natural bitterness of the fruit by degrading oleuropein. Also, it will reduce the antimicrobial activity of the phenolic components which are present in the olive fruit. Also, the NaOH treatment will make the skin of the olive more permeable to allow the sugar diffusion to improve the fermentation. After NaOH treatment, the olives are washed and brined in 10% NaCl for fermentation. Initially, there will be a variety of gram-positive bacteria and gram-negative bacteria. And as a result of their growth, organic acids will accumulate. And as the pH decreases below 6, their growth will get inhibited. And the lactic acid bacteria such as Lactobacillus plantarum and Leuconostoc misandroids dominate. And there will be yeast species such as Candida, Pichia, Saccharomyces, etc. The growth of these microorganisms will contribute the desirable flavor characteristics to the fermented olive. Next olive fermentation is the natural black olives. These are prepared by a slow fermentation without lye, without sodium hydroxide. For this, Black olives are picked in a ripened state. They will have a black color and as well as they will be having softer texture than the green olives. Since there is no sodium hydroxide treatment, the fermentation will be slower. This is due to the fact that there are the presence of antimicrobial phenolic compounds and there is no diffusion of sugars, which will slow down the fermentation. Generally, the microflora associated with natural black olives are Saccharomyces, Hansenula, Candida, Torulopsis, Diberiomyces, Pichia, Pleuropomyces, Cryptococcus, etc. The next olive fermentation is ripe black olives. These are prepared by darkening olives through oxidation. So there is no initial alkali treatment. The ripe black olives are prepared from green or semi-ripened olives and they are brined without an initial sodium hydroxide treatment. They will be directly kept in a brine solution for up to 1 year and after that the olives will be subjected to oxidation treatments in the presence of 1 to 2% sodium hydroxide which will blacken the olive. They are then washed with water to remove sodium hydroxide and then will be canned in 1 to 3% sodium chloride brine. The next fermented vegetable is Sunki. This is a non-salted fermented radish product traditionally consumed in some northeastern state of India, Nepal and Bhutan. The fermentation is carried out by Lactobacillus plantarum or Lactobacillus brevis. For the production of Sunki, fresh radish roots are washed and wilted in sunlight for 1 to 2 days. Then they are shredded, washed and packed in a jar and kept for fermentation at 30 degrees Celsius for about 12 days. During this period, Lactobacillus fermentum and Lactobacillus brevis and Lactobacillus plantarum will grow and their growth will decrease the pH to about 3.3. After the period of fermentation, the product is sun-dried to remove the moisture content and then it will be undergoing a processing in a clay-lined pit for 2 to 3 months. The next fermented vegetable is gundruk. This is a non-salted fermented vegetable made from the leafy vegetables such as mustard, cauliflower, radish, etc. This is used in Nepal. For the production of gundruk, shredded leaves of the above said vegetables are packed in an earthen pot with warm water for about 5 to 7 days. During fermentation, Pediococcus pendusius, Lactobacillus cellulosus and Lactobacillus plantarum will grow. After fermentation, the product is sun-dried to remove the moisture content. The next fermented vegetable product is Sunki, which is a non-salted fermented vegetable prepared from the leaves of Otaki turnip. This is generally used in Japan. For the production of Sunki, the Otaki turnip is boiled and mixed with a wild small apple and a dried portion of Sunki from the previous year. This mixture will be kept for fermentation for about 1 to 2 months at a low temperature, generally during winter season. During the fermentation, Lactobacillus brevis, Lactobacillus plantarum, Bacillus coagulans, Pediococcus pendusius, etc. will be growing. The next fermented vegetable is Kawal, which is a fermented leaf preparation prepared from the Wild African Legume Cassia obtusifolia. This is a highly rich in protein product and is generally used as a substitute for meat. For the production of Kawal, the leaves are not washed. They are ground into a paste and kept in an earthenware pot for about 14 days. During this period, the contents will be mixed once in 3 days. The microorganisms involved are Bacillus subtilis, Propionibacterium, Lactobacillus plantarum, Candida krusei, Saccharomyces, Rhizopus, etc. Now we will discuss cucumber fermentations. This is also known as pickling. The word pickle usually refers to a pickled cucumber. The cucumber pickles may be prepared unfermented or partially fermented or fully fermented. For cucumber fermentation, the cucumber or Cucumis sativus is harvested before mature. Mature cucumbers are generally not fermented since they are large, they will ripen easily and they are full of mature seeds. Cucumber fermentations typically undergo a homolactic acid fermentation by Lactobacillus plantarum and the other related lactic acid bacteria. Generally, there is no production of carbon dioxide. There are two types of cucumber fermentations: salt or salt stock pickle and dill pickle. The salt or salt stock pickle. Here, immature cucumbers are washed, packed in barrels or tanks and brined. Sometimes 1% glucose is added. Depending upon the level of salt, two methods are there: high salt method and low salt method. In the high salt method, 10.5% brine is added initially and 9 pounds of salt per 100-pound cucumber are added. In the low salt method, a low amount of salt is added initially, that is 8% brine is added initially and 9 pound salt is added per 100-pound cucumber. During the primary fermentation for about 2 to 3 days, most of the unwanted bacteria will disappear and the lactic acid bacteria and yeast will proliferate. Initially, there will be Pseudomonas, Flavobacterium, Bacillus, Leuconostoc Streptococcus faecalis, etc. And during the final stages, after 10 to 14 days, Lactobacillus plantarum and Lactobacillus brevis followed by Pediococcus will predominate. The traditional process takes 6 to 9 weeks for completion. The commercial cucumber fermentations are commonly done in 30,000 to 40,000 liter open-top fiberglass tanks and they are generally exposed to sunlight. The UV radiation in sunlight will kill the aerobic surface yeast, which will otherwise decrease the level of lactic acid by metabolizing it. After fermentation, the fermented cucumbers are washed to remove the excess salt and then they are packed with a cover liquor which may contain acetic acid and spices as well as the residual lactic acid. Further microbial growth and spoilage in a fermented cucumber is prevented by the presence of organic acids, the low pH and the lack of fermentable sugars. Next is dill pickle. These are named so because they are flavored by the addition of a herb known as dill and also some other spices like garlic and onion may also be added. Here, a lower concentration of salt is used and the brine is generally acidified by using vinegar. The low salt content allows more microbial production of acids. There are two types of dill pickles: overnight dill pickle and genuine dill pickle. In overnight dill pickle, it is prepared by slow acid fermentation at a low temperature in a low acidified brine. So here the brined cucumbers are held at 3.3 degrees Celsius where they undergo a lactic acid fermentation at a slower rate and as a result a 0.3 to 0.6% acid is developed. Due to the low content of acid, overnight dill pickle will be having a shorter keeping time. Next is genuine dill pickle. It is prepared by using a brine which contains 7.5 to 8.5% salt. The temperature during fermentation is between 15 to 30 degrees Celsius. The final acidity in the product varies from 1 to 1.5% and the concentration of salt in the finished pickle will be between 3.5 and 4.5%. Now we will discuss various defects or spoilages that may happen in a pickle. They are shriveling, which result from the physical damage due to the presence of higher concentrations of salt, vinegar or sugar in the solution. Next is hollow pickles, which are formed when the cucumbers are left to stand for some time after harvesting and before fermenting. Floaters or bloaters in a pickle results when there is a growth of heterofermentative lactic acid bacteria, which will produce carbon dioxide during the fermentation. So the fermented cucumbers will be having undesired internal gas pockets. Next is slippery pickles. This occurs when the cucumbers are exposed to air due to the growth of capsulated bacteria. Next is soft pickles. These are due to the result of pectinolytic enzymes, which may come either from the growth of mold or it may come from cucumber flowers. The next defect is black pickles. This develop due to the formation of hydrogen sulfide by bacteria. Next is ropey pickle brine, and this is due to the growth of certain gram-negative capsulated bacteria. So we were discussing about fermented vegetables. We discussed about fermented cabbage, fermented olives, different fermented leafy vegetables and fermented cucumber. Thank you so much for listening.